Let's see here. Which one are we looking for? This one. So we are now at week 4 of term 5. We are almost halfway to the end of the school year already. So because we are at the midpoint of the term, that means that our midterm exam is coming up. So a midterm exam is a big exam that covers all the stuff that I've talked about in class so far. And the way that I like to do my midterms is that I post them on Wednesday and then give you guys until Friday in order to complete it. So here it'll be posted tomorrow morning, just like a normal assignment would be. But unlike a daily assignment, it is not due until the end of the day on Friday. So I'll talk about it in a little bit more detail at the end of class today. I just wanted to bring it up now just to kind of mention that it is coming up. It is a bit longer than a normal assignment. It covers everything that I've talked about in class so far, from the DNA replication to the protein synthesis, and even including some of the stuff about cell division that we're going to be going over today. And so I'll give you guys a little bit of a look at it at the end of class today, but I just wanted to mention that it is coming up this week right here at the top, just to kind of help make you guys aware of that. All right. So going into our notes for today. So we are going to be covering cell division this week, which is what happens when cells grow and split into 2 or more new cells. It's the way that all the cells in your body are made, by cells growing and dividing and growing and dividing in order to create new cells. And so today we're going to be talking about what's happening with the DNA inside of your cells, what's kind of the way that it's packaged when it comes to getting ready for this division. And we'll start to get to, or we'll get through basic cell division, which is called mitosis. And then on Thursday, we'll continue on with cell division. We'll talk about the other type of cell division, which is a little bit more specialized version called meiosis. So here is our DNA double helix spiral that we've been talking about so much the last couple of weeks. Again, we've got the genes in our DNA that are responsible for making those proteins. But the DNA is not just floating around inside of the nucleus of our cells. We have a ton of DNA, and so it really needs to get packaged up tight. And the way DNA gets packaged is into these big structures called chromosomes. So the first part of today is kind of be about what these chromosomes are and how they're structured inside of human cells. So the basic idea is that we take all this DNA, we wrap it up really, really tight, we string it around these kind of bead-looking things that are called histones, and then we squish all those guys together into the chromosome. It's a way to basically cram a whole lot of DNA into a very, very tight, very small package. So the entirety of a cell's DNA, all of the DNA in your cells are called its genome. So genome is just an easy way to refer to the entire DNA strand, not just these genes, not just G's genes, not just this part of DNA, but all of your DNA is your genome. And like we've been talking about last week, the DNA is so important because it has all of the instructions for how to build the proteins that make your cells work. So everything that your cells need to survive and function is based on proteins. And all of the information to make those proteins is in the DNA. We take the gene, we copy it into the mRNA, and then the mRNA goes to the ribosome and uses those amino acids to make our proteins. So each cell, if you took your DNA and like put it all into one big, long, super tiny, thin string, there are about 3 meters, which is about 9 feet of DNA in every single one of the millions of cells that make up your body. So obviously, we need to take that 9 feet of DNA and squish it down really, really tight and small in order to get it to fit in your cells. So the way we do this is we package the DNA into chromosomes. And the basic way that chromosomes work is that there's not just one chromosome in your cells. It's not just one huge long strand of DNA, but instead there are multiple chromosomes. So your DNA is basically separated out into these multiple sections, and each section is called a chromosome. So that's a big long section that gets condensed down into that tight little package. So here is kind of like a cartoon view of what our chromosomes look like. You may notice that each set of chromosome comes in a pair. So in humans, we have 23 pairs of chromosomes, and we'll talk about that more in a second. And each pair is called a homologous chromosome, or homologous chromosomes. So homologous just means that they're the same chromosome. So we have 2 copies of chromosome number 1, and 2 copies of chromosome number 2, and 2 copies of chromosome number 3, and so on and so forth. And each copy of your chromosome has the same basic genes on it. So each copy of 1 has the same set of genes. Each copy of 2 has the same set of genes, so on and so forth. The reason we have 2 is because in humans, we have 2 parents. So in each one of our homologous chromosomes, one of the copies you get comes from your mom, and one of the copies you get comes from your dad. So that's why you essentially have 2 full sets of DNA in each cell. One set of copies from your mom, and one set of copies from your dad. So that's what makes these homologous chromosomes, also sometimes called homologous pairs, is we have these pairs of chromosomes, one from your mom and one from your dad. So all of your DNA, all the DNA that makes you is condensed down into 23 pairs of chromosomes that are inside every single cell in your body. So every single cell has the same copies of these 23 pairs. Whether you're a skin cell or a muscle cell or a brain cell or a bone cell, every single cell has the exact same set of 23 copies of chromosomes. So a pair is called homologous chromosomes, and you get 1 from each parent. Like I was saying before, each pair is identical except for the X and Y chromosomes. So in humans, whether you're born as a male or female is determined by these chromosomes right here. If you're born with XX chromosomes, then you are a female. And if you're born with XY chromosomes, then you're born as a male. So these ones can be different. The Y chromosome is a lot different from the X chromosome. However, all other 22 copies are all have the same sets of genes on them. So the homologous chromosomes contain genes for the same traits. So like on this pair of chromosomes, loci is just a fancy science way of meaning like the position on the chromosome. And so for this case, say this is the gene for eye color. So this pair of chromosomes both have the eye color gene on them. So depending on the combination of eye color genes you get will determine what color eyes that you grow up to have. So this gene, this is what makes that protein that'll determine, in this case, your eye color. And let's pretend that it's chromosome 7. I don't know for sure. But let's just say that each copy of chromosome 7 that you have, both have eye color genes, both in the same place. So the interaction between those two genes, and depending on what gene you get from which parent, determines what type of traits you'll end up having. And we'll talk a lot more about that next week or the week after. I can't remember. I think it's next week we get really into genetics and how these genes work. So we have our homologous chromosomes, 2 copies of the same chromosome. Each copy you get from a parent, and the copies have the same sets of genes on them. So a karyotype is just a fancy kind of picture of all of the chromosomes. So like here is an actual human karyotype for a human male. Somebody went through and took all the chromosomes out of the cell, lined them up together, and took pictures of each pair. Again, in humans, we have 22 pairs of normal chromosomes plus 1 pair of sex chromosomes. So 23 pairs in total. And again, this is a human male, which is why we have XY for our sex chromosomes. Fun fact about sex chromosomes, whenever you're having a baby, the sex of the baby is completely determined by the dad. So since women are XX chromosomes, they can only give X chromosomes to the baby. So you will always have an X chromosome from your mom, but then whether or not you get an X chromosome or a Y chromosome from your dad, that is what determines if you are born as a male or a female. So you always will have 1 X from the mom, and then if you get an X from the dad, it'll be born as a girl, and if you get a Y from the dad, it'll be born as a boy. Yeah, I thought so too. All right, so normal everyday activity of a cell, it just has 2 copies of each chromosome, 1 from the mom and 1 from the dad. But then that second week of class, we talked about DNA replication, right? We had the leading strand and the lagging strand, and the helicase opened it up, and then the primases were coming in, and the polymerases were putting down the new DNA, all that good stuff that you definitely 100% remember for the midterm. Well, when we make a copy of our DNA, we make a copy of each chromosome. So we call the copied chromosomes chromatids or sister chromatids sometimes. So like I said, you start out with a copy from your mom and your dad. So let's say this is your mom's copied. We then replicate your mom's copy and make an exact duplicate. So now at the end of DNA replication, you have 2 copies from your mom and 2 copies from your dad, the one that you inherited and the one that you made in your cell. And we call those copied chromosomes sister chromatids. So there's a lot of kind of confusing terminology about chromosomes and chromatids, which is why I like to go over it right here at the beginning. So the homologous chromosomes are the mom and dad copy that are normally in your cells, and the chromatids are what you get when you duplicate those copies. And so when you're getting ready to divide a cell and the DNA copies and replicates, you'll have 2 chromatids from your mom's chromosome and 2 chromatids of your dad's chromosome. So you end up with 4 total copies of each chromosome, the one that you started with and the one that you made a duplicate of. All right, I know there's a lot of terminology that I'm throwing at you guys right now. So does anybody have any questions? Anybody still a little bit confused about chromosome versus chromatid type stuff?
No.
All right, great, Naya, thanks. Water before we keep going. All right. So your DNA is packaged up into chromosomes, which are different segments. You start off with 2 copies of each segment, the homologous chromosomes, the one from your mom and the one from your dad. And then after you go through DNA replication and you're getting ready to divide the cell, you copy the chromosomes and now you have chromatids. So you have double copies of your mom's chromosome and double copies of your dad's chromosomes. So chromosome number can vary quite a bit between species. It's not correlated at all with like species intelligence or evolutionary advancedness. It all just kind of depends on how that species developed. So in our normal body cells, which in science and biology, we call them somatic cells. So a somatic cell is like 99% of the cells in your body. So your skin cells, your brain cells, your hair cells, your muscle cells, these are all somatic cells. And so the number of chromosomes in there is known as the diploid number, or 2n. Diploid meaning essentially double, because again, in normal cells, we have 2 copies of every chromosome, 1 mom copy and 1 dad copy. So when you have the normal mom and dad copies, the normal homologous pairs of chromosomes, we call that the diploid number, or sometimes you'll see it referred to as the 2n number, meaning like the doubled chromosome number. So that's true for 99% of the cells in your body. They are somatic diploid cells. They have 2 copies of every chromosome. The other 1% of cells are known as gametes. And these are specifically the sex cells. So in females, for humans, it's eggs. For males, it's sperm. These guys are a little bit different because they only contain half the number of chromosomes, known as the haploid number. So they don't have the homologous pairs going on in these haploid cells, also sometimes called just or represented as N. So N means you have 1 copy of all the chromosomes. 2N means you have 2 copies of all the chromosomes. Again, most cells are diploid or 2N, most cells have the mom and dad copied together, those homologous chromosomes together. But your gametes, your sex cells, your sperm cells, and egg cells, they only have the haploid number. They have only one copy of each chromosome. So instead of having 2 copies of number 7 and 2 copies of number 13 and 2 copies of number 20, they just have the one copy.
Oh, actually, I was gonna say, it's because I'm writing it down and you get the last part. So can I write just really quick?
Sure. Go for it. So yeah, diploid means 2 copies of every chromosome. This is like the most common state for 99% of the cells in your body. Haploid only occurs in the gametes, only occurs in the sex cells. And in haploid cells, we only have one copy of every chromosome. So, thanks, Ashley. So N means you just have the single number of copies. 2N means doubled up copies. 2N goes with our diploid cells. Single N goes with our haploid cells. So species have all sorts of different chromosome numbers. And this is our diploid numbers. This is the total amount of chromosomes that are in a cell from this species. And again, more or fewer chromosomes does not mean more or less advanced. It can be totally random. It all depends on how the species evolved. So like camels, for example, have a total of 70 chromosomes in their cells. Squirrels have 40 chromosomes. Chickens have 78. King crab have a whopping 208 chromosomes in their cells. Fruit flies are a lot more simple. They have 8 chromosomes. Tomato plants have 24 different chromosomes. Potato plants have 48 different chromosomes. And finally, we as humans, our diploid number is 46. In 99% of your cells, there are a total of 46 chromosomes. One set of 23 from your mom, and one set of 23 from your dad. So our haploid number in humans is 23. This is how many different chromosomes that humans have. 22 regular chromosomes plus the sex chromosomes. So in a haploid cell and a sperm cell or an egg cell, you will find a total of 23 chromosomes. However, if you looked in like a cheek cell or an arm muscle cell or a brain cell, you would find 46 chromosomes. 23 chromosomes that you inherited from your mom and 23 that you inherited from your dad coming together to give you a total of 46. So that's how many are in a normal diploid cell with the doubled up chromosomes from your mom and your dad. However, there are 23 different chromosomes. So 23 homologous pairs in a diploid cell gives us 46. It's really, really, really important that you have exactly 46 chromosomes in your cells. There are a lot of different conditions and diseases that are caused by people having even just one more or one fewer chromosome in their cells. So in a lot of cases, if something happens and you don't end up having the right number of chromosomes, then that human won't even develop. So this is actually what's responsible for a lot of miscarriages where people initially think they're pregnant, but then they end up losing it very early on. That's because as the cell is developing, it notices that it doesn't have the right chromosome number. For whatever reason, when the sperm and egg came together and fertilized, not the right number of chromosomes were in there. So that's what causes it to stop growing and eventually be passed out, is because it immediately shuts everything down once it realizes not all of the chromosome numbers are right. So that doesn't always happen. There are a lot of cases of people that are born with not the correct number of chromosomes. But if that number is too far off, that's what will cause the cells to shut down. So in some cases, you could have monosomy, which is like you have one copy, meaning you're missing a chromosome somewhere. Instead of having 2 copies of number 7, you only have 1. So that causes you to have 45 chromosomes instead of 46. You could also have trisomy, which is having an extra chromosome. So trisomy, like 3. So this is where you end up with a 3rd copy of a chromosome. Instead of having 2 copies of chromosome 21, you end up having 3 copies of chromosome 21. So you end up with 47 chromosomes instead of 46. So the reason that this happens is essentially just because sometimes when the chromosomes are getting split up from cell division, which we'll talk about later on, they don't get split up evenly. They're supposed to get split up so one goes to either side of the cell, but sometimes they don't get separated, and so you end up with too many chromosomes in one gamete and not enough chromosomes in the other gamete. So that has a fancy science name called non-disjunction. So this is usually what's responsible for people ending up with the incorrect chromosome number. These sister chromatids or homologous pairs don't break apart evenly, they don't separate correctly, and so you end up producing cells that have the wrong chromosome number in them. Specifically, your gametes, like your sperm and egg cells. So here we have another human karyotype. Remember, a karyotype is just a fancy picture of chromosomes. Can anybody see what is wrong with this karyotype? Is there something that's off about this particular chromosome picture?
Number 21 has 3 instead of 2.
Exactly. Ashley has spotted it right away. If you look down here, they all have 2 copies, 2 copies, 2 copies, 2 copies over here. But we have 3 sets of chromosomes for number 21. So this is not a extremely rare condition. It's called trisomy 21. It's like what's happening at the chromosome level. But does anybody know the condition that is caused by trisomy 21, by somebody having 3 copies of chromosome number 21? Oops. So trisomy 21 is also known as Down syndrome. So you guys have probably seen people with Down syndrome before. They have the altered facial characteristics. Usually their eyes are a little bit wider and a little bit further apart. And they have learning difficulties and mental disabilities. All of those conditions happen. All of those physical and mental conditions happen just because they inherited an extra chromosome when they were developing. So this is why cells go through such effort in order to make sure that you end up with the exact correct number of chromosomes. Because even just having one more chromosomes. This person has 47 chromosomes instead of 46, but that is enough to have all of these host of issues in terms of physical and mental abilities. So chromosomes, extremely important, and your cells work really, really hard in order to make sure that you end up with the correct amount. All right. Anybody have any more questions so far about chromosomes, chromosome number, haploid versus diploid, anything that you're still a little confused about or want some more clarification on? All righty. So the reason why I focus on chromosomes so much right at the top of this presentation is because chromosomes play a very big role in cell division. The process of taking one cell and growing it and splitting it apart into new cells. So at one point in your lifetime, you were a single fertilized egg cell inside of your mom. And then that egg cell grew and divided and grew and divided and did that a couple million more times until there's enough cells for you to be born as a baby. And then as you grew up and got older, all the cells in your body grow and divide and grow and divide in order for you to age and mature and grow and turn into the person that you are today. And even as an adult, you still have cells in your body that are growing and dividing all the time. The average blood cell only lives for a couple weeks before it has to be replaced. The average skin cell only goes for a couple months before it has to be replaced. And so you're constantly making new skin cells, new blood cells, and making new cells to repair any sort of cuts or bruises or damage that you get inside your body as well. So cell division is an extremely important process that's happening all the time inside of your body. So we need to repair broken or damaged cells or replace dead cells, and that's why we need cell division. We want there to always be healthy cells inside of our body. And the way we do that is by replacing the ones that get injured or that break down and die. It's also really important because, like I said, at some point you start off as a fertilized egg inside your mom. And in biology, we call that a zygote when it first starts growing and developing. And all of the cells in your body come from that initial zygote, that initial fertilized egg. So it's not only important to repair or replace cells, we also need it in order to grow, right? There are a lot more cells inside of you now than there were when you were first born as a baby. And that's all thanks to cell division. So I talked about the cell cycle a little bit when we were going over DNA replication. And you may remember that the main thing that I focused on then is that the S phase is where that happens. The S phase is the synthesis phase, and that is where we make our DNA. But all the phases are important, and all together they're known as the cell cycle. And you can kind of think of this as like the life cycle of a cell. So it starts as a newly formed, newly divided cell, and then it grows, and then it copies its DNA, and then it grows some more, and then it splits up to become new cells. So there are kind of 5 stages that you can think about the cell cycle growing. So as soon as a cell is produced, it enters G1 phase. G just stands for growth. So G1 is sort of your basic growth phase of the cell. It's getting bigger. It's producing its proteins. It's doing whatever job it needs to in the body, whether it's a bone cell or a muscle cell or what it might be. This is kind of normal average growth of the cell. So all cells go through this phase during their lifetime. Most cells, a lot of cells at some point or another, will then realize that they need to divide in order to make new cells. So the first step to do that is we then, after G1, enter our S phase. And remember, we already talked about this guy. This is where we replicate our DNA. We make a copy of all the DNA in our cells. So this is where we go from just having the homologous chromosomes, the mom and dad chromosome copied together, and we make those chromatids, those doubled chromosomes, now that we have copied our entire DNA. So after we copy the DNA, we're still not 100% ready to split right away. We then have to go through the G2 cycle. So G2 is where we're getting ready for cell division. So mitosis, we'll talk about a whole lot here in just a second. It's basically like regular, normal cell division. So in G2 phase, the cell is kind of getting ready for that and making sure that all the pieces are in place. It's making sure that the organelles, the little structures inside of the cells, are ready to be copied as well. Finally, once we're all good to go after G2, we have the M phase, M for mitosis. This is where we actually copy the cell. We take all the DNA, we split it to opposite ends of the cell, and we split the cell nucleus apart. We make sure that it's all separated out. And finally, the last phase is called the C phase. Sometimes, depending on who you talk to, some people will lump this in with the M phase. So the M and C happen really, really close together. C stands for cytokinesis, which is just a fancy science way of saying that the cell pinches apart and becomes two new cells. So at the end of the C phase, we have two brand new cells from the original cell that we started with. So again, right after the C phase happens, both of those new cells will then enter the G1 phase. So we do our initial basic growth. We copy our DNA. We get ready for cell division and copy the organelles. We then go through cell division very quickly, split the cells apart into two new cells, and we go back into our G1 phase. So these three phases together, you'll hear a lot of time, they're usually grouped together in what's called interphase. And interphase is just a way of saying the cell whenever it's not actively dividing. So it's growing, it's getting ready to divide, but it is only actively dividing and going through the division process down here in the M and C stages. These three together, we're just kind of getting ready for that moment. We're getting ready to divide, especially with the S and G2 phase. So here, I like this illustration for it a lot. We have our brand new cell. We enter the G1 phase. The cell will spend most of its life going through this, growing and developing and becoming the best cell that it can be. Eventually, it'll realize that it has to get ready to go through cell division. And that's where we enter the S phase, where the DNA gets copied. So we take those homologous chromosomes and make those sister chromatids. We then, after the DNA is copied, we then make sure that everything else in the cell is getting copied and everything's getting ready to divide. And then finally, we very quickly go through mitosis, separate all the parts to the different sides of the cell, go through cytokinesis, split those cells apart, and they become two brand new cells, both of which will then immediately go into the G1 phase. So mitosis and cytokinesis happen very, very quickly. It's a very fast process. So most of the life is in this interphase section. So we spend a lot of time going through G1. It takes us a long time to copy the DNA and to get all the other stuff ready. And then we very quickly take the DNA and separate it and cut the cell, and now we've got new cells. So that's sort of our basic life cycle of the cell. It grows, it copies everything, it separates apart and gets split into two, and then those two new cells start growing again. So there are a bunch of different reasons why cells will go through cell division. A lot of cells will do it if they just get too big. So at some point in a cell's life, if it gets really, really big, then it's not very efficient at moving the stuff around the inside of the cell. And so it'll divide itself to make two smaller cells that can do the job very well. Sometimes, especially if there's like certain cells that we really need, or like if you have a cut on your skin and you need to build some new skin cells right in that area, there will be like growth factors like hormones or chemicals released by other cells that will tell the cell like, hey, you need to start dividing like right now. Start copying that DNA. Let's go. And so sometimes it happens kind of naturally. Sometimes cells are encouraged, or cells, it's kind of the body demands that they start dividing as soon as possible. But the important thing is that we're going to copy our cell, and we're going to make new copies of this body cell. So before we move on and start talking about exactly how the cell goes through the process of separating its parts and splitting apart, does anybody have any questions so far? Anything about the chromosomes or the cell cycle that you have questions about, or you're still a little bit confused on? All right. So I've been talking about cell division as just kind of this very general, basic type of thing that we need to do. But there's actually two different types of cell division. The first type, the one that we're going to focus on in kind of the second half of class today, is mitosis. Mitosis is sort of like your standard, normal, everyday cell division. So this is the division that like 99% of the cells in your body go through mitosis. So mitosis produces identical cells. At the end of mitosis, both of the cells that you've made are identical, not just to each other, but also to the original cell that you started with. So if you start off with a blood cell, you go through mitosis, and now you have two exact copies of that blood cell. So this occurs in our body cells, in our somatic cells that we were talking about earlier. Again, 99% of your body is made up of these somatic cells, and those are what go through mitosis. The other type of cell division that we're going to talk about on Thursday is a lot more specific to certain cell types. But mitosis is what 99% of your body cells go through. The cells that you end up with in mitosis are diploid cells. Again, our body cells, our somatic cells, are diploid, and we're making identical copies. So if our original cell is diploid, our newly created cells are going to be diploid as well. So our final results of cell division with mitosis are going to have two identical cells that both have two copies of every chromosome. So we'll talk in a lot more detail about exactly how mitosis works, but essentially we start off with our homologous chromosomes that make us diploid. So we have two copies of the same chromosome. We go through replication. So now we have those chromatids. So each chromosome got duplicated. We then go through mitosis in order to separate those chromosomes apart. And now we end up with two diploid cells. They both have two copies of each chromosome, and they are both identical to the cell that we started with, and to each other as well. So that's mitosis, and today we're going to talk about exactly what it's called when we go through these different steps to separate out our chromosomes. Mitosis, normal, every day, happens all the time. This is what creates your new skin cells, your new bone cells, your new brain cells, your new blood cells. They all come out of mitosis. So again, we start off with one cell that has the copied chromosomes. Those guys get separated and split apart to opposite ends of the cell. And then eventually we'll snip the cells apart and they'll become two identical cells, two identical diploid cells. So mitosis happens in stages, right? So there are these like sequence of events that we have to go through in order for the cells to be properly separated apart. And they're called, sometimes they're called stages of mitosis, sometimes they're called phases of mitosis, but it's basically sort of like the recipe for how to correctly separate a cell. So our first stage, or our first phase of mitosis is called prophase. This is like, once we've gone through all the other parts of the cell cycle, this is when things start to get real and we start to get prepped for mitosis. So I like to remember prophase is the first part of mitosis because this is where we're getting prepped. This is where we're getting ready to go. So the chromosomes bunch up really tight. Before, you can't really see the chromosomes in the cells, but when we're about to go through mitosis, they became very thick and very apparent as the DNA gets super tight in with each other. And our nucleus breaks down. So the nucleus is that part of the cell where all the DNA and all the chromosomes are hanging out. But obviously, if we're about to pull the cell apart, that nucleus can't be holding all that stuff in. So in prophase, the nucleus starts to disappear. We also have things called asters and spindle fibers appear. So asters are usually represented by like these little block looking kind of things. And basically asters and spindle fibers are going to be the things that actually physically pull the chromosomes, pull the DNA apart. And so in prophase, they're going to appear and they're going to kind of start shifting over and getting ready to be in place. The whole end goal is to separate the DNA, separate the chromosomes onto each side of the original cell. So the asters are going to start moving to opposite ends of the cell to get ready to pull that DNA. So our spindle fibers are like the ropes that are actually going to latch onto the chromosomes and pull them apart. So the asters are sort of like these anchors on either side. And then the spindle fibers are the ropes that kind of get extended out, and then in a couple stages, they're going to pull apart. So again, prophase is our prep phase. This is where we're all getting ready to go through mitosis. Stuff starts to appear, stuff starts to disappear. Everything is starting to be put into place. That's prophase. So here in late prophase, you can see these guys going through. Here's our chromosomes. And then here is our spindle fibers, which are just starting to attach on to the middle parts of the chromosomes. They're just starting to get ready for that pull. Up next, we have metaphase. And I always found metaphase really easy to remember because metaphase is the middle phase. In metaphase, the chromosomes are going to line up right on the equator of the cell. So like you can see, they're all lined up right here. And then in this one, they're all lined up like this. But in metaphase, all the chromosomes kind of come and line up with each other right along the middle part of the cell. So this is where those spindle fibers are really important. They're what are really connecting in and starting to line all the chromosomes up correctly. They attach on to the center part of the chromosome, which is called the centromere. And so metaphase, middle phase, everything's getting lined up. Everything's getting ready to go. So now you can see our nucleus is completely gone. Our asters, our anchors, are here on opposite sides of the cell, and all the little fibers are reaching into the middle, attaching to the chromosomes, and lining it up in a straight line, right up and down or right across the middle part of the cell. So if prophase is the prep phase, metaphase is the middle phase. So if you ever see an image like this or a diagram like this where you have all of the chromosomes all lined up really close to each other, all making a pretty straight line through the cell, that's a good signal that you are in metaphase. The next step is the big, probably most important stage of mitosis. It is called anaphase, which I always remember as the action phase. So if metaphase is the middle phase, anaphase is the action phase. And I call it the action phase because this is where the magic happens. Those spindle fibers pull to either side of the cell and separate the chromatids from each other. So one half of our chromosomes is coming over to this side. One half of our chromosomes is going over to this side. So these asters are pulling the chromosomes to opposite sides of the cell, and it's the spindle fibers that are actually doing the attaching and the physical pulling. So anaphase is the action phase. Basically, as soon as metaphase is done, as soon as all of those chromosomes are lined up, they're pulled and separated apart in anaphase. So whenever you see chromosomes that are like in the middle of being separated, of being pulled apart, that's how you know you are in anaphase. So we've got the chromosomes separated from each other. They're on opposite ends of the cell. And now it's time to start finishing things up, start kind of tidying things up. So the next stage of mitosis is telophase or telophase, depends on how you want to say it. Basically, now that the chromosomes are on the opposite sides, they can relax a little bit. They don't have to be packed in so tight. They become a little bit looser. And probably most importantly, the new nucleus starts to appear. So in telophase, now that the chromosomes are all separated, you've got the new nucleus that starts to come in and surround the chromosomes. Because remember, the chromosomes are our DNA, and the cells do not like it when the DNA is just floating out there naked and unprotected. So as soon as these guys get separated in anaphase, we start immediately making that new nucleus. And so that means we are in telophase. So we're not 100% done yet. We've separated the DNA, we've separated the two sides of the cell, but we're still connected right here, kind of in the middle part, right? There's still this little tiny bit of cell membrane that's connected to each other. So once we've gone through metaphase and anaphase and now telophase, where we're closing everything off, we have to do a final little separation of the cell to make it fully be two new cells. And that process is called cytokinesis. Some people put this as a phase of mitosis. Some people say, no, it's not part of mitosis. It happens afterwards. Regardless, all that really happens is that we fully divide the cytoplasm, the inside of the cell. We fully break the cells apart and make two new, what are called daughter cells. So in cell division, any new cells that are created from the original cell, they're called daughter cells. And cytokinesis is where the two fully formed daughter cells show up. Again, some people say this is part of mitosis, and some people say, no, it's separate, it's after. I don't really care which is which. The main thing is just to remember that cytokinesis is just like that final tiny little step where we pinch apart the cells to create two fully formed, brand new daughter cells. And remember, we're in mitosis, so each cell is the exact same, and each one is an identical copy of the original cell, what's called the parent cell. So telophase, we separated the DNA, we start to make a new nucleus, and the DNA starts to relax a little bit, but it's not until cytokinesis that we actually fully chop apart and separate the cells. So here is a nice little diagram. Again, we're in prophase. So our nucleus is starting to disappear, and we've got our chromosomes that are bunching up and becoming very dark and very obvious. Oh, actually, I was doing that wrong. So here is our original parent cell. We've got two blue chromosomes and two red chromosomes. Oops. We then go into prophase. The nucleus starts to disappear. The DNA is doubled up and super tight in close to each other. Those asters and spindles start to form on opposite sides. Metaphase, they all line up right up and down the middle. Right here, the spindle fibers are attaching and hooking together. Anaphase, the action phase. We pull the chromosomes apart. One half of the chromatids ends up on this side. The other half of the chromatids ends up on this side. And then in telophase, we start to reform the nucleus. Eventually, it will go through cytokinesis and snip the cells apart from each other. But we end up, after we're done in mitosis, with two identical daughter cells. So each of these daughter cells has two blue and two red chromosomes, the exact same as each other, and the exact same as our parent cell up here. And this guy has two copies of each chromosome. These guys have two copies of each chromosome because they are diploid. So mitosis produces new diploid cells. Each one of these daughter cells has the exact same sets of DNA as the parent cell that it started from. So again, regular, standard cell division. You can almost think of this as like making a Xerox copy of the original cell. You make a copy and now you end up with two new cells that are exactly the same. So 99% of the cells in your body go through this process, or at least have gone through this process at some point in their lifetime. So prophase, we're preparing. Metaphase, we're in the middle. Anaphase is the action phase. And then telophase brings it to a close, where we start to reform all the stuff that was gone before. All right. Anybody have any mitosis questions? Anything about these phases here that you're still a little confused about? All right. So again, at the end of the day with mitosis, it's all about making two identical cells that are diploid and have the exact same diploid set of DNA as our original parent cell. So I always try to remember it as mitosis is 2. So you've got like the 2 right here in the middle. So mitosis makes 2 cells, and those 2 cells are the exact same as the original cell. So here we have some different cells which are in different stages of mitosis. We have a pretty clear anaphase up here. Most of these guys are just regular cells that have this nucleus right here. This guy looks like it's about ready for metaphase, getting pretty close to lining up in the middle. These guys look like they're starting to get ready for telophase. So this one and this one are now separated, and they're starting to reform their nucleus. So these are actual pictures of cells where the DNA has been dyed so you can see it more easily. But I think these are from like an onion root or something like that. So just a really thin shaving of cells that were put on the microscope and were pictured while certain cells were going through mitosis. Again, prophase, we start to break down that nucleus, and all the chromosomes become condensed and those spindles start to form. Metaphase, they all kind of line up with each other across the middle, and all those spindle fibers are really attaching. Anaphase, the action phase, they get pulled apart, totally separated. And then telophase, we start to reform the nucleus and start to separate the cells from each other. And then they'll be finished off by cytokinesis. All right, so on Thursday, we'll get into meiosis, which is a different type of cell division where we do not end up with 2 cells at the end. It does not happen in most body cells, and the cells that we end up with are not identical to each other. So we'll get into exactly why that is and exactly how meiosis works when we come back together on Thursday. So your daily assignments for today and tomorrow are going to focus pretty much just in on this mitosis aspect of things. So today's assignment is just some questions about chromosomes and the cell cycle. So the first part, I just want you to answer some basic questions about what chromosomes are, what are the multiple different copies that we have, and why do we have them? And then for the second part, I want you to tell me which one of these descriptions matches with which part of the cell cycle. So for these ones, it'll just be, there's not multiple choices for each answer. Each description fits with just one phase of the cell cycle. So pretty simple overall. 11 questions, but most of them are just this matching part down here. So that's the assignment that got posted for today. And then tomorrow's assignment has to do with those mitosis phases that I was talking about earlier. It starts off with a little bit of a review just to kind of remind you of what they are, since this will be for tomorrow. And then a little parts about interphase, which is that kind of in-between phase when we're going through the regular cell cycle. And then our preparation prophase, our middle metaphase, our action anaphase, and our reforming step of telophase. And so these questions down here, the answers for the most part are either going to be one of these phases or some information that you can find about them that is in the little write-up from each one or from the beginning up here. So all of the answers to all these questions down here, you can look back in your notes if you want, but they'll all also be in these different sections of the top part right here. So questions about things like interphase and prophase and metaphase, what's happening in them, what are kind of the main steps going on in these different phases. And again, that'll be the daily assignment that gets posted tomorrow. So make sure to log on and complete it in order to earn your Wednesday attendance. All right, any more questions about the homework for today and tomorrow, or anything about the notes that I've gone over so far?
No.
All righty. So the last thing that I wanted to remind you guys about, I talked about it a little bit earlier, is the midterm that's coming up this week. We are about halfway through our term 5 so far. So the midterm exam is going to be posted tomorrow morning, but you have until Friday evening to work on it, the evening of the 30th. So you have all day Wednesday, all day Thursday, and all day Friday to work on it. It is an open note exam. So you have all of the notes that I've posted in the Google Classroom so far to go back to and reference if you need help on some of the questions on the midterm. It is a little bit longer than a normal homework assignment, which is why I'm giving you a couple days to do it. I think it's somewhere between like 20 and 25 questions. And it's made up of a wide variety of questions like multiple choice, short answer, fill in the blank, matching, stuff like that. So if you do it on the Google Docs, it'll look just like doing a normal exam on paper. The Google Forms are a little bit different than the other ones, just because since it is a really long exam, I broke it up into sections so you don't have to keep continuously scrolling through the whole thing. And so like the first section right here just has, you put in your name and some directions, and then once you answer or put in your name, you can hit next. And then, like I said, there's some multiple choice questions. There's some matching questions with our DNA. There's some short answer questions and fill in the blanks. And then you'll answer all these guys, hit next, and it'll take you to the next page where it has another set of questions. You'll go through all of those until you finally hit the submit button at the very end. Again, it is a little bit longer than a normal homework assignment. And so I definitely recommend taking a look at it and working on it starting tomorrow, if at all possible. But again, it is not due until Friday. And some of the stuff that we cover this week, including, actually, I can't remember if there's any meiosis stuff on the midterm, but there could be some cell division questions on there as well. So everything from DNA replication, protein synthesis, the mRNA, the ribosomes, the amino acids, all that stuff is covered on the midterm. So again, it'll be available to start looking at and start working on early tomorrow morning, but it is not due until the end of the day on Friday. So you have all day Wednesday, all day Thursday, and all day Friday to look on it, work on it, and get it turned in. Definitely make sure to read all the questions carefully. And if you do get stuck, you can always go back to the posted notes in the Google Classroom to help you. If you are ever unsure of what one of the questions is asking or how you're supposed to answer it, you can always reach out to me as well. Obviously, I can't help you answer the question itself, but if you're stuck on it and you're kind of confused about what you're supposed to do for it or how you're supposed to answer it, that's the type of stuff that I can help you out with. I don't want anybody not getting something correct just because they didn't understand how to answer the question. So please reach out to me with any questions that you have on it, and I'll do my best to help you out as much as I can. All right, any questions about the midterm exam that's going up this week?